the regulation of IP3-induced calcium release. So it's a genuine pleasure to have you with us, Ian. And I'll, without any further delay, hand over to you.
Okay, great. Right, thank you, Martin. So you can see the screen okay and hear me okay?
Absolutely fine, yes. Looks good.
Great. Okay, so thanks to you and Katya for the invitation to present. So I'm speaking from COVID isolation on a bright sunny morning in California. So my talk is going to be based on a paper that we had out in eLife just a few weeks ago. As the title says, we're looking at IP3-mediated calcium release during global spikes. And we've gone back and re-examined something we thought we got sorted out over 2 decades ago. In fact, revising hypothesis that my lab had put forward a long time ago. So to begin, some ancient history. History of calcium signaling, calcium imaging. And as imaging techniques developed, one of the first discoveries was that individual cells show oscillations in calcium level, repetitive spikes, seen in many different cell types, thought to be a sort of frequency encoding of information to regulate numerous cellular processes. So at first, it just looked as if calcium simply went up and down homogeneously in a cell. But as the techniques developed, it became apparent that each calcium spike was often seen as a wave that spreads across a cell. And back some 20 years or more ago, my lab discovered things, calcium puffs, little local calcium transients. Here we're just recording from a small part of a xenopus calcium signals in response to IP3. You see these little calcium volcanoes going off, things we call calcium puffs. One at the corner here, and she triggers a wave that then spreads across this part of the oocyte. So calcium puffs, a little local signal, which seems it can act as a trigger to propagate waves. On the trace here, you can see these are calcium spikes preceded by calcium puffs, and then we get a much bigger global rise in calcium. And our hypothesis for what was going on to generate these signals, local signals and then a global signal, is a dual function of the spatial arrangement of IP3 receptors in a cell and the properties of the IP3 receptor channels that liberate calcium from the ER. The IP3 receptors are biphasically modulated by calcium. Particularly low levels of cytosolic calcium promote the opening of channels. So our picture is that IP3 receptors are grouped in clusters along the ER membrane. One channel may open, give a tiny calcium signal we call a PLIP, but that release of a little bit of calcium from this channel could trigger openings of adjacent channels. So a puff results from concerted opening of a handful of clustered IP3 receptor channels. And then at higher concentrations of IP3, we can get a propagating global signal, calcium released from one local cluster. It can diffuse, activate the next cluster, that diffuses, activating the next. We get a saltatory propagation of a calcium wave that gives a global signal. So on that idea, we're thinking that calcium puffs are the universal building block of a global calcium signal. The global signal is the concerted activation of many puff sites. So we had originally proposed that idea now more than 20 years ago, and it's become well accepted. As one example, a recent paper from Colin Taylor's lab in the title. They say puffs, the universal building blocks of IP3 evoked signals. But although there's been many papers that have looked at the transition from puffs to the global spike, how puffs may trigger a global response, it's actually been difficult to figure out what's going on during the global spike itself. Is it really the summated activity of numerous puffs? And the problem there is simply that during the global rise, calcium levels rise high, the fluorescent signal goes high. It becomes difficult to resolve individual puffs, what individual puff sites are doing. So we set out to re-examine that hypothesis. And to do that, to try and look at what the puffs might be doing during a big global signal, we came up with an image processing method of fluctuation analysis, to try to better resolve what's happening with puffs during a global signal. So let me just say a little bit about the techniques we're using. So it's image processing. We do calcium imaging. We're now doing it with turf imaging. On what I'll be showing you is cultured HEK cells loaded with Cal520 using caged IP3 to photorelease and stimulate. So here, movie I'll show you, this is the raw fluorescence record from the cell, what would come out of the camera. And we then process these records through an algorithm that's looking for fluctuations. On a pixel by pixel level, we're looking for changes in time of the fluorescence. The calcium puffs are very brief, transient local signals. The global rise is a much slower, spatially homogeneous. So by doing this analysis, fluctuation analysis, we can separate out the puffs better. And what we get, we call it a standard deviation signal. It's a running measure of the standard deviation of the fluorescent signal's local calcium changes at each pixel in the cell. So let me play this video. There's quite a bit going on, so I'll play it a couple of times. To begin with, just look at the two panels on the left. We'll begin with baseline. Partway through the record, we photorelease IP3 to evoke a global response. Let me start that running. Here's baseline. Now we photorelease. The playback slowed a little. You can see the calcium is rising here. And on the trace, it's showing our SD signal. Let me just play that one more time. As we photorelease, you can see little spots appearing here, the puffs. They're just about visible in the raw record, but hard to make out. But our SD signal picks them out beautifully. And then on the right, this is just showing a plot from these two records. Measurements of the raw fluorescence, that's the cyan trace, and the global measure of the SD signal is putting a region of interest around the entire cell, looking at an average of all these local fluctuations. So that's the yellow trace. And this is indicating puff activity throughout the cell during this response. And what you can see is there's a flurry of puffs early on during the rise of the calcium. The puffs terminate, calcium rise continues for a while, and then it's sustained, coming down slowly, even in the presence of uptake sequestration mechanisms, which would otherwise bring it down much more rapidly if there weren't a sustained release going on. So that's our method. Statistically, we can get a measure of puff activity from these fluctuations. This is just a control to convince you that this method is valid. Doing the same thing, HEK cell. This time, instead of stimulating with IP3, we're stimulating by putting on a low dose of ionomycin to cause a gradual release of calcium from organelles. So let me play this video. Again, the ionomycin produces a rise in the global calcium. The SD signal, we're not seeing these little hotspots, no puff activity. Looking at the traces, we have a rise in calcium, comparable in amplitude to what I showed you with the IP3, but the SD signal is flat, no puff activity. So just showing you some illustrative results. Again, here the smooth traces are all just the raw calcium signal from a cell. The noisy traces are ST signal representing puff activity. Top three, we just photorelease increasing amounts of IP3. Small amount, we have puffs that continue for a long time, only a very small rise in global calcium. More calcium, we get a faster rise in the global signal. And again, we see the puff activity. Its kinetics follow. When we give a strong photorelease of IP3, we get a fast rise in global signal. Again, we get this transient flurry of puff activity, which then very quickly ceases as the global calcium rises and stays up. Most of the time, we're using photorelease of IP3 as our stimulus. Bottom trace just to show that this mimics what we get with agonist activation, here applying carbachol to activate the cell's own IP3 signaling pathway. So, this is our observation. One question we had was, why is this puff activity terminating? Why do we just see this brief flurry during the rising phase of the calcium signal? And let me just stay with this one for a second. You can see from these traces that the kinetics of the puff activity and the rise of the calcium seem to match. As we get a faster rise in global calcium, this flurry of puff activity gets shorter. So it seems the puff activity is in some way tracking the rise in global calcium. And to look at that, what we did was just take measurements at each time point on the rising phase. We said, what is the global fluorescence, a measure of the global calcium? What is our standard deviation at that same time, a measure of puff activity? And then we can do a scatter plot of one against the other. So here the horizontal axis is global calcium level at a given time. Vertical axis, what is the puff activity at that same time when the global calcium level is at that particular point during the rising phase. So here are the data for traces I showed you before. We divide them up as slow, medium, and fast rising signals. They all seem to follow the same curve. If we just replot these data, but now normalize the same peak, they all lie on pretty much the same relationship. So our first thought was, maybe the puff activity is being determined by the global bulk calcium level. Maybe the puffs are terminating as the calcium level goes high. Maybe what we're seeing is a reflection of the classic bell-shaped curve for the calcium dependence of IP3R channel activation. Increasing calcium levels at first increase the probability of opening of IP3 channels, and then as the calcium goes higher, that probability goes down. The channels tend to shut. So is this bell-shaped curve the same as that bell-shaped curve? Are our puffs terminating simply because calcium is becoming high during the global signal? So we set out to test that. To give you the quick answer, it seems to be no. It's not elevation of global calcium that's terminating the puff activity. And one experiment we did to test that, shown here. Just look at the top left-hand panel to begin with. Here's a response to carbachol, global rise in calcium, RST signal, the puff activity. So this is very much what I was showing you before. But what we're doing in this experiment, we loaded the cell with caged calcium, so we could then give a preceding UV flash, elevate the basal calcium level before we apply the carbachol. So then if you just look at the right-hand panel here, at this point we photorelease calcium, we give a jump in basal calcium level from the starting point here up to the arrow where our trace begins again here. Now we've got this big elevation of calcium in the cytosol. We put the carbachol, we again see a global IP3 evoked signal, and we still see the SD signal. Both are somewhat reduced. There may be some inhibition going on, but if we just plot the ratio of how big is the global signal to how big is the SD signal, which we're doing here, for different amplitudes of the preceding calcium jump, essentially that remains constant. Elevating the calcium level before we photo or evoke an IP3 evoked response is not suppressing the puffs. So we're concluding our puffs are not terminating because of a rising global calcium. So what might be the explanation? So we then thought, well, calcium in the cell is pretty much a zero-sum game. The calcium which is going up in the cytosol, it's coming from the ER, so the ER calcium levels are dropping. Is it a drop in the ER calcium which is terminating the puff activity? So here's an experiment to test that idea. Let me talk you through this top trace. Again, the smooth trace is global calcium level fluorescence. Noisy trace is our SD signal reflecting puff activity. Our cells bathe in zero calcium solution, so we're not getting any calcium influx across the membrane. We apply some CPA transiently to inhibit Circ activity. We have a leak of calcium from the ER, we get a rise in cytosolic calcium. We wash out the CPA, wait for a while, and then we photorelease IP3. And what you see now is we've still got a global IP3 evoked calcium release, but there's nothing on the SD signal. We have a calcium release mediated by IP3, but without any puff activity underlying it. And you can see this enlarged on this trace. In this case, we've kept our cell bathed in zero calcium, so the ER couldn't refill. Global response, but no puff activity. As a control, we repeated this exact same procedure, but now with calcium in the bath, so the cell had a chance to refill during this time. Now, when we gave the UV flash photorelease IP3, we have our usual response. We have a global signal, but we recovered our puff activity. So our conclusion is that we have a flurry of puff activity during a global response, but it quickly terminates because of a partial depletion of the ER calcium. So it looks like two different, functionally different sources of calcium. Calcium released during puffs and calcium coming from some other source. And we're calling those a punctate release mode involving the puffs, and then a diffuse mode, rising calcium evoked by IP3, diffuse in time, slowly rising, diffuse in space, we can't see local signals associated with it. One thought was, if we have these two different modes, could it be different isoforms of IP3 receptors that are mediating these two modes? The answer there is no. All three isoforms seem to mediate both modes of release. So here we're using HEK cells engineered by David Ewell, cells which express exclusively type 1, type 2, or type 3 IP3 receptors. Here are typical responses to carbachol. In all cases, there are differences in amplitudes and kinetics, but fundamentally the qualitative conclusion, both or all three IP3R isoforms mediated both puffs, this flurry of puff activity, the SD signal, and the global rise. Another possibility we thought about, could our diffuse signal be arising from puff sites in the cell interior? Everything I've shown you up to now was done by TIRF imaging, so we're only looking at calcium right next to the plasma membrane of the cell. And using light sheet imaging, we found, particularly in the HEK cells, that we can see puffs arising in the cell interior. So here again, this is just raw calcium fluorescence Here's our SD signal reflecting puff activity. But now these are light sheet images. We've taken a diagonal slice through the interior of the cell. And as you can see, we have a lot of puffs around near the plasma membrane. That's where most puff sites are, but there are still several inside the cell giving puffs. So could it be that these interior sites keep on going for a while, diffuse calcium from them diffuses slowly to give us what looks like a diffuse signal at the plasma membrane? And the answer again seems to be no. The puff sites in the cell interior show very much the same behavior as those on the periphery around the cell membrane. So in these traces, again, plotting SD signal, puff activity, global calcium rise. In this case, the blue signals were recorded from region of interest in the cell interior, the red around the periphery. They're both doing very much the same thing. Puff sites in the cell interior show the same kinetics as those around the periphery. And on average, the signals we're getting from the cell interior are appreciably smaller than from the peripheral puff sites. Okay, last thing we looked at was just how important are the puffs in terms of the total calcium release underlying the global signal. So this plot is a little bit complex. Let me talk you through it. To begin with, the black trace is just global calcium fluorescence, global calcium signal throughout the entire cell. The grey noisy trace, again, is our SD signal, puff activity. So we have this brief flurry of puffs during the rising phase of the global calcium signal. We then tried to estimate what is the calcium flux into the cytosol. We did that by taking the rise in the global calcium, what calcium is in the cytosol, and then from that we subtracted what we estimated the calcium sequestration to be. In separate experiments, we gave a pulse of cytosolic calcium, looked at the reuptake to get a measure of the sequestration rate. And then from the global signal and our estimated the sequestration rate, we could get a red trace here showing our estimate of calcium efflux into the cytosol. So this has an initial spike, but then a very maintained component. Our puffs are only going on for a short time at the very beginning of the signal. Just this initial spike of calcium efflux. After the puffs cease, there's a continuing efflux, and we just integrated under the red curve to get a measure of the cumulative calcium release. How many moles of calcium have been released during this entire response? That's the blue curve there. So it's pretty obvious that the puffs have ceased by about this time, so they're contributing only a small fraction of the total calcium released. So we estimate something like only 20% or so of the total calcium released during the global response is coming from the puffs, and the rest would be coming from what we're terming this second diffuse release mechanism. Okay, so to summarize, take-home message from my talk, we believe there are two distinct modes of IP3-mediated calcium liberation during global spikes, punctate and diffuse. Under normal conditions, both mechanisms are going on. We have a flurry of puff activity during the rising phase, but then it's sustained by a diffuse mechanism. And we can separate these two components. Middle panel here. If we apply some EGTA into a cell, which buffers the global rise, and give a relatively low photorelease of IP3, we can get puffs which continue for a long time. They're picked up very well by our SD signal, with very little or no global rise in calcium. If we partially deplete the ER, then we can still have there's enough calcium left to get a global rise, but our SD signal is flat, no puffs. So clear discrimination, punctate release, diffuse release. So that, at the moment, that's as far as we've got. We have many questions. Some relate to the generation of these two modes of calcium signal. Exactly why do the puffs terminate during this flurry? It seems to be a local depletion of calcium or a depletion of ER calcium. Is it a local depletion due to the puffs themselves? Is it a more global ER depletion by the diffuse release? Why are the puffs terminating? Is it just a reduced driving force for calcium flux across the ER as the ER levels drop? Or is there some modulation of the IP3R gating? And then what we're seeing is two different modes of calcium release. But does that imply two different populations of IP3Rs? Might it be that the IP3Rs that give the puffs, their gating is being modulated, so instead of giving puffs, they're just giving some sort of very gradual trickle of calcium? Or is there a completely separate population of IP3Rs giving the diffuse release? So that's the generation of the signal. Then a whole load of questions in terms of downstream signaling. Puffs are a local release. Effectors, organelles close to the puff sites will experience transient large rises in calcium. The diffuse release appears to be uniform through the cell at a lower concentration. That will represent a very different signal. So then how are all the different effectors, channels, calmodulin, organelles, and so on, how are they located in relation to these different modes of calcium release? How do they respond to the different kinetics of the punctate versus release? Okay, so we have those questions. We're just starting to try to answer them, but then we got shut down by COVID. So these are still questions. And to finish, let me just acknowledge funding sources and the people who did the work. These are my lab group members. Jeff here essentially did all the experimental work and contributed greatly to the analysis, the writing of the paper. Kyle was involved in writing the software we're using. Divya, a separate paper, we developed the theory behind the fluctuation analysis method we're using. Okay, so thank you for your support and welcome if you have any questions of your own.
Thanks very much, Ian. That's really super interesting. Can you hear me okay?
Yes, I can.
Great. So we do have some questions. Maybe I can just kick off with one. So just interested in the termination of the calcium puffs you see during a global response. So the idea is that you get a calcium puff and inside the ER you get kind of like an anti-puff, to borrow something from the Spark nomenclature. And that's a local depletion that then terminates that puff. So what a couple of things I'm puzzled about here is during the global response that continues for many tens of seconds, as you've shown in some of your recordings, why doesn't ER calcium tunneling allow that puff to fire again? And there are certainly many situations where, because it would restore that anti-puff to normal or pretty much normal calcium level, you would have thought, because those people who have recorded ER luminal calcium levels during calcium oscillations have shown that it's actually quite a modest depletion. And in addition, in some cells that we've recorded calcium puffs from, you can see the same site firing again with quite high frequency sometimes, which suggests that they can actually repeat fire for the entire time course that you would be recording a global signal. Now, of course, maybe there isn't sufficient depletion there to shut them down, but certainly it suggests that if there is an anti-puff going on in the ER, it's not enough to cause a long-term inhibition, which is what you're seeing. I just wonder if you can answer that.
Well, my quick answer would be no, I can't.
Okay.
Elaborate a little bit. One thing, there are two different mechanisms going on. that a puff itself terminates within 100 milliseconds or so. We and Colin have argued that isn't a local depletion of calcium that terminates an individual puff, because we can see single channel calcium signals, they don't get smaller. What's going on during the global signal? It seems to be repeated puffs at a relatively high frequency. That's associated with the termination of the puff flurry. Whether that is the cause, I don't know. And you made some good arguments against it being a local depletion by the puffs themselves. But maybe it's still a lot of puffs, several channels at a cluster, repeated activity. Maybe there could be a local depletion at that region of the ER. But as you argue, I think that possibility is not so likely, not so probable. But what we'd really like to do is try and get some good ER calcium imaging going to get a direct test.
Yeah, that would be fascinating. Yeah. Okay, so a question coming in from Kevin. I think probably a lot of us are thinking the same one. So Kevin says, in nuclear patch clamp electrophysiology, inhibition of IP3 receptors requires very high micromolar calcium. So in your uncaging experiments, you might imagine that global calcium levels that you achieve wouldn't be the same as those that that would inhibit IP3 receptors, perhaps, when they're activated by an agonist. So does it?
Let me just try to figure out the context of the question. And so the experiment we did with is that in terms of the caged IP3 experiment?
Yeah, I'm pretty sure that's where Kevin's question was coming from.
Yeah. Okay, so the caged calcium experiment, we gave calcium elevations that in terms of global delta F over F naught level were higher than when we'd see complete suppression of puffs during IP3 evoked responses. So we tried to match the calcium levels, that initial step of calcium, the full release of caged calcium took us to a level at which during the rising phase of an IP3 evoked signal, the puff activity would be essentially completely suppressed.
Okay, thinking that that would mimic the calcium-dependent inhibition you would get from the IP3 receptors themselves feeding negatively back.
Yes, at least in terms of a global cell average calcium level, yes.
Okay. Okay. We could pick that up again later on, maybe. Okay, from Colin, Ian, any thoughts on whether the loss of noise with global signals arises from recruitment of different lone IP3 receptors, or loss of coordinated activity within the clusters of IP3 receptors that evoke puffs, e.g. uncoupling caused by low luminal calcium or high local cytosolic calcium?
Okay, so if I understood that correctly, Colin is suggesting that could the diffuse component be just individual IP3 channels just going off randomly by themselves with no coordination?
Right.
And
or do the puffs become uncoordinated?
The puffs become uncoordinated. We think that's not the case because in the imaging, we can very clearly resolve what we believe to be single IP3R channel openings. They give us an SD signal. When we look at the peak of an IP3 evoked response after the puffs have terminated, when we look at the partially depletion, when we have a global rise, but without any apparent puffs, we see essentially no fluctuation noise. We're now trying to look very carefully at that. We're doing power spectrum analysis. We're really not seeing any excess noise. I think it's very clear that if it was coming from uncoordinated individual channels opening with the regular conductance with a regular calcium flux, we would readily be able to resolve that.
Okay, so following on that kind of theme, so Martin Falk is asked, does the flurry of calcium puffs trigger the diffuse mode of release in control situations? Could we imagine just the diffuse mode as physiological signals? Do you have to go through puffs to get to diffuse mode, or can you engage the diffuse mode by itself ever?
Well, I think clearly, and we'd shown that prior partial depletion of the ER, we can have the diffuse mode without any puffs. Whether that's physiological is another question. And one thought that comes to mind is that sometimes you probably came across cells that just don't give puffs very well.
Absolutely, yes.
Responses, we tend to just, in the past, we certainly say, oh, that's a bad batch of cells, let's get some new ones. That may be more of a pathological than a physiological condition. I think the answer is certainly you can get a diffuse mode release without the puffs, but it does look as if the puffs may be playing an important role in triggering the global response, in accelerating the initial rise of the global signal, maybe in helping propagate a calcium wave.
Yeah, we certainly did see cells that didn't give calcium puffs. We presumed that we were just out of focus, but we may need to think about that differently. So have you considered the implications of IP3 receptor density on different modes? Have you played around with that at all? There's a question from Joseph Heiss. Does local IP3 receptor density influence punctate versus
density in terms of simply
like
total
clustering
number
or not?
of per cell,
Yeah.
or whether it's a clustered or diffuse. And in terms of the spatial arrangement, clustered, diffuse, then clearly, I think it's well accepted now that the puffs are arising as a result of the clustered arrangement of the IP3Rs. There are good grounds, experimental and theoretical, for thinking that this clustered arrangement enhances the calcium release from a given number of channels because they're much more able to coordinate by being close to one another. In terms of our diffuse mode release, an attractive possibility is that it's coming from a large fraction of IP3Rs in a cell that are not enclustered. As Colin's shown, a larger part of the IP3Rs in a cell, unlike the puff sites, they are motile in the cell. They appear to be functionally silent, certainly under conditions that give puffs. Possibility is that that population of IP3Rs could be mediating the diffuse release. But at the moment, we have no evidence one way or the other. Right. While it might still be the same IP3Rs at the clusters, at the puff site clusters, but they just switched to some very different mode of calcium release, that is trickling calcium.
Mm-hmm. Interesting.
Just to throw it out, maybe a slightly wild hypothesis, we wonder if the diffuse release, maybe it isn't actually so diffuse. Maybe it's coming from nanodomains, perhaps those communicating with mitochondria. Mm-hmm. And what we see as the diffuse release is just calcium trickling from the edges of those nanodomains and filling up the bulk cytosol. That we're not seeing nanodomains, we're just seeing leakage from nanodomains. But anyway, that's a sort of interesting possibility that we're just playing with.
Do you think it's possible, actually, that it could be calcium going into mitochondria during puffs and coming back out of mitochondria in a less spatially constrained a way? That, if, for example, just as a way of it leaking out, not just out of a MAM situation, but also out of out of mitochondria, could disperse a calcium signal?
I think not, and most directly, we just pre-treated cells with FCCP. Mm-hmm. And then looked at the IP3 evoked signals. We see that very little change. We still see this pattern of punctate puff release and a global diffuse signal.
I see. Okay, interesting. Okay, and finally, I think before we take a break, Holly Smith has come in with a question about the different physiological roles of these two sources of calcium. Where would you go with that in your mind?
Where would we go? Okay, so the immediate thoughts are to look under conditions where we can selectively activate just puffs, do some EGTA loading, relatively low IP3, and then using this partial depletion method to just evoke a diffuse release, and then to look at some downstream effectors. What is happening to mitochondrial calcium signals, to try to get a readout of gene expression. Can we see selectively different downstream effects from punctate versus diffuse release?
Yeah, fascinating. Okay, well, there are a few more questions, but we are running quite a long time over at the moment. So I think what I'll do is say thank you so much, Ian. Really interesting presentation. We don't intend to run off. So if people want